Okay, hi everyone, and in this module we're going to discuss some topics in genetics. So we're going to cover initially studying DNA, RNA, the molecular components of genes, and then we're going to go on and talk a bit about some definitions and some terms we use when we're discussing genetics. And then we'll follow that up with some inheritance patterns and how you can calculate inheritance patterns in certain conditions. Okay, so it's an interesting topic. It may be a bit confusing and take some time to digest it, but that's like most things in life. It just takes some time to learn it, and then once you start to understand it, it'll become more and more interesting. Okay, so let's follow this sequence from the base pairs, which is on the right, all the way to the left, where we have a cell with a nucleus in it that is containing the genetic information in the form of chromosomes. Okay? And we have 23 pairs of chromosomes. We'll discuss that a bit later, which actually makes 46 chromosomes in total. Okay? And remember, the chromosomes are the genetic information of the body. And we now have completed the complete genetic mapping of human DNA. Okay? So let's go to the right side. Let's look at base pairs. So base pairs are the building blocks of DNA. Okay? And they're made up of guanine, which is a nucleotide, cytosine, thymine, and adenine. And you'll see on the right there that guanine attaches to cytosine, thymine attaches to adenine, and so on and so on. And this will continue for thousands and thousands of base pairs. And that's what makes up DNA, right? Deoxyribonucleic acid. And we'll get into a bit more detail in an upcoming slide on what a nucleotide looks like. Okay, so now we have our DNA, which can continue for many, many thousands of base pairs. And that's going to make up a chromosome, right? And chromosomes are going to have many, many different locations on them that are going to code for different information. And we're going to call those different sections alleles. And we'll come back to that. Okay? So different parts of a chromosome will be responsible for specific genetic information that can then be coded for and produced within the cell. Okay, we'll come to that process. Okay, so it goes base pairs. The base pairs make DNA. The DNA is actually what the entire chromosome is made of, basically. And then where do all the chromosomes exist? Within the nucleus. And there's 23 pairs of chromosomes. Okay, again, in this slide, it's just showing you the sequence from a gene. Okay, well, what's a gene? It is a basic unit of hereditary. So it's a segment of DNA that's going to exist on a part of a chromosome, right? And there's going to be hundreds and hundreds and hundreds of genes that will code for specific information that can then be expressed as a protein or some kind of genetic expression in the body. Okay, so a gene is a segment of DNA Where is it? It's on the chromosome. Where's the chromosome? Within the nucleus of the cell. Okay? And remember, DNA is a double helix. It's this amazing double helix twisting molecule where adenine attaches with thymine and cytosine attaches with guanine. Okay? And you're going to see later in this module that if we have gene defects, that can impact characteristics of that particular person. Okay, so we'll talk about different gene defects a bit later. Okay, so on this slide, we're just looking at the structure of a nucleotide. So in one of the above slides, we saw base pairs, say adenine, thymine, cytosine, or guanine, which are all the ones that are in DNA. We'll talk about uracil in a minute. And what do these nucleotides look like? Well, they have a phosphate group. And remember, phosphate typically likes to live on the outside of structures, right? Because it's hydrophilic, such as the cell membrane. Okay, so we have a phosphate group in DNA or RNA. We're talking about nucleotides right now. And then we have a sugar group, which could be ribose or deoxyribose, right? Ribose in RNA, deoxyribose in DNA. And then finally, we have one of these nucleobases. So it could be adenine, it could be thymine, cytosine, or guanine in DNA. In RNA, it's slightly different, okay? We don't have thymine in RNA. So adenine pairs up with uracil, and cytosine pairs up with guanine, which is the same as DNA, okay? So what's a nucleotide? It's a phosphate group attached to a sugar group attached to one of these bases. Okay, so let's go into a bit more detail about bases. Okay, so purine bases, in pyrimidine bases, in DNA, and also in RNA. Okay, so remember, DNA is deoxyribonucleic acid, and RNA is ribonucleic acid. Why do I say that? Because we're just describing it based on the fact that it's deoxyribose, just the type of sugar it has. And nucleic acid, that's referring to the nucleic acid portion of it, which could be adenine, could be guanine, could be thymine. Okay, RNA, ribonucleic acid, because what's the sugar? It's a ribose sugar. So ribonucleic acid. Okay, so what about the different bases? So we can have pyrimidine bases or purine bases. And as we've seen in DNA, which is a double helix structure, adenine attaches to thymine and guanine attaches to cytosine. The difference in RNA is that there is no thymine. RNA uses uracil as its base instead. Okay, so let's look at the bottom here. So adenine attaches to thymine. They pair together in DNA. A attaches to uracil in RNA to make a pair. And cytosine and guanine always attach together in DNA or in RNA. Okay, so let's look at DNA versus RNA. Where is DNA? Well, it's in the nucleus of the cell, and that's where it stays. Okay. What is it made of? It's made of a double-stranded helix with base pairs of adenine attaching with thymine and guanine attaching with cytosine. over and over and over again for thousands and thousands of base pairs. Okay? RNA, usually single-stranded. Okay, so RNA can be a copy of DNA within the nucleus and then travel to the cytoplasm. And we'll get to that on another slide. So what's adenine made of? What's RNA made of? Well, what are the bases? Well, adenine, guanine, cytosine, or uracil. Notice that thymine doesn't exist in RNA. Okay, so here is the lovely structure of DNA, right? So this image is going to help you see that the phosphate groups are attached to a sugar group, which is then attached to a base, okay? And the base can be thymine, cytosine, adenine, or guanine. And it becomes a double helix because thymine attaches with adenine and cytosine attaches with, to guanine. And if we look at a certain section of DNA strands which are on the chromosome, we can call that a gene. Okay, and the gene will code for a certain protein. So we'll go through that process in upcoming slide. But just know the composition of nucleotides and how they pair together in DNA and which ones are in RNA. Now, in the top right-hand corner, it's going to remind you, yes, there's a phosphate group. Yep, there's a sugar group. In the case of DNA, it's deoxyribose. And yes, there's a nitrogen base, and that nitrogen base can be A, T, C, or G. Okay? So in actual fact, DNA or the bases are nitrogen based. Okay. Okay, so the DNA that is living within the nucleus in the form of chromosomes can go through a process or perform a process called transcription. And it can code and make a messenger RNA. Okay, so, and this is an important process because the DNA wants to get its genetic information to the cytoplasm in the form of mRNA so that it can be copied into a certain protein. Okay, so the DNA is the genetic coding of every cell in our body. And in actual fact, the genome of every cell in your body is identical. So the cell on the end of your nose and the cell at the end of your foot has the identical DNA. It has the identical genome. And what differs is which genes are turned on, which are activated, and which aren't being used. Okay? Because only a portion of the DNA is being used for protein synthesis. Okay, so that's why liver cells can look different than cells on the end of our nose, because different parts of the DNA are activated and making different proteins. But in actual fact, the genome of a liver cell is the same as the genome of the cell at the end of your nose. Okay? So just know that the genome is always the same in every cell. It just depends which parts of the genome are activated. Okay? So, we're going to talk next about how this process occurs from DNA to messenger RNA and then into the cytoplasm. Okay? And remember, A goes with T, and G goes with C, as base pairs of DNA. Okay, so RNA, so ribonucleic acid. So there's three types. But what's the main role of all of them, all three types? It's to take the information in DNA and turn it into a protein that can be used in the body, right? Because we have so many different types of proteins that perform different functions. And where do these proteins get coded for? Well, the information is initially within the DNA. Okay, so we have mRNA, which is messenger RNA, and this is going to be basically a copy from the DNA, right? It's going to be transcribed right off the DNA in the nucleus. And this is going to carry instructions from the nucleus into the cytoplasm. Okay? Then we're going to have what's called transfer RNA, which is going to be the RNA that carries the particular amino acids that are going to code for a particular protein. And there's a good image coming up. So let's just go through the three types and then we'll go to the image. Lastly, there's ribosomal RNA. The ribosomes actually have RNA within them. Okay, we're not going to go into the detail of exactly what ribosomal RNA is, but we'll see in the upcoming image of how each three of them interact with each other. Okay, so let's start at the top right where you see the nucleus. And you can see the messenger RNA, which was copied off of the DNA in a process called transcription, has now come out into the cytoplasm. Okay, messenger RNA. And it's just a long single strand of nucleotides that have just been copied from the information that was on the DNA. So if, for example, there was thymine on the DNA, well, that became adenine on the RNA. If there was adenine on the DNA, it became uracil on the RNA, and so on and so on, with this long strand of messenger RNA. Okay, so the messenger RNA is now floating around in the cytoplasm. It's going to migrate to the endoplasmic reticulum, where the ribosomes will then attach onto the mRNA. Okay, and there's ribosomal RNA within the ribosomes. Okay, but another important aspect of this is the transfer RNA. So the transfer RNA has amino acids attached to it. And the particular amino acid that's attached to the transfer RNA depends on the three base codon. Okay, so for example, you'll see at the bottom AUG on the mRNA. Okay, on the transfer RNA, there's going to have to be UAC, and that's going to code for a particular amino acid. And what makes up a protein? It's a long strand of amino acids. So there's an infinite types of proteins, because it just depends on the sequence of which amino acids have been put one by one into making the protein. Okay, and that is totally dependent on that codon that was on the mRNA. Okay, so you can get an endless array of proteins based on what that codon on the mRNA was coding for. Okay, so we did the overall process there. Let's go back into the nucleus. Okay, transcription. Transcription is the process of making a copy of messenger RNA from the DNA. Okay, so the DNA is copied into mRNA by an enzyme. And this process is called transcription within the nucleus. Remember, the DNA never leaves the nucleus. It needs a messenger. And that messenger is the long strand or the short strand of messenger RNA that then goes into the cytoplasm to attach with ribosomes. Okay, so the process of making proteins is called translation. So we just spoke about transcription. That's the process of making mRNA from DNA. Okay. Now what's the process of making protein from mRNA, which is in the cytoplasm, actually on the endoplasmic reticulum, which is an organelle. And here is a lovely image showing you how the protein, or if it's short, we'll call it a peptide, is produced, right? So at the bottom in pink, you have your messenger RNA. You have the ribosomes that are attaching onto the messenger RNA. So there's a large unit of the ribosome and a small unit that attaches on, and now it's going to code. It's going to code the mRNA. It's going to go along the mRNA, and it's going to look at 3 base pairs and decide which amino acid is next to attach to the developing protein. Okay, it's a pretty amazing process. All right, messenger RNA, ribosomal RNA attaching to the messenger RNA, and then the transfer RNA bringing in a particular amino acid that's going to be attached to the strand of a peptide or a protein that's developing. Pretty amazing. Okay, so let's just go through this together because we just spoke about transcription. Okay, that's taking place in the nucleus. That's the production of mRNA. Translation, okay, that's taking place in the cytoplasm, and that's the process of the production of protein. Let's go back to transcription. Okay, so the first statement, product of transcription is mRNA. Yes, it's being copied off the DNA that becomes mRNA. Transcription occurs in the nucleus. Yes, for sure. Okay. DNA never leaves the nucleus. Transcription requires RNA polymerase. Okay. RNA polymerase is just an enzyme that is transcripting. It's looking at the DNA code and it's making a copy of mRNA for it. Okay, let's go over to translation. The product of translation is a polypeptide, that's just a protein, that eventually becomes a protein. Okay? Translation occurs in the ribosomes. Well, translation occurs typically on the endoplasmic reticulum with the ribosomes attached to the mRNA. Okay, the bottom one. Translation uses various reagents to create a polypeptide chain. Well, translation is using the mRNA, it's using the ribosomes, it's using the transfer RNA to bring in individual amino acids. So I think that's what they're referring to there. Okay, so there's a lot of moving parts to copying that mRNA and turning it into a protein. Okay, let's go in the middle now. So both cytosine and guanine. Both have cytosine and guanine. Well, that's true. DNA has cytosine and guanine, and RNA has cytosine and guanine. Adenine and uracil as their nucleotides. Well, adenine and uracil are nucleotides for RNA. Adenine and thymine are nucleotides for DNA. Okay. Transcription needs a template. Yes, the template is the DNA. Translation needs a template. Yes, translation's template is the mRNA. Okay, so hopefully you understand the difference between transcription, that's DNA to mRNA, translation, mRNA to protein production. Okay, we don't need to spend too much time on this at all. There's the DNA on the left in the nucleus, the process of transcription, which is making a copy of the DNA in the form of messenger RNA, which floats into the cytoplasm and will get decoded in the process called translation and make, as a final product, a protein. Okay, so let's do this question together. Which type of RNA moves from the nucleus into the cytoplasm carrying the genetic code? Messenger RNA, that's the answer, right? So messenger RNA in the process of transcription gets produced in the nucleus and moves into the cytoplasm. Transfer RNA is the RNA that's carrying in the individual amino acid to attach onto the developing peptide or protein. Okay. Ribosomal RNA, no, that's RNA that's within the ribosomes already. They don't carry information from the nucleus to the cytoplasm. Okay. All of the above? No, that one's incorrect too. So it's messenger RNA.